Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. I hope you had a great week. Like always, remember that you can find Let's Talk Micro on Apple Podcasts, Amazon Music, Spotify, Google Podcasts, Stitcher, TuneIn Radio, Overcast, Pandora. Wherever you listen to your podcast, you can find Let's Talk Micro. I am also on Instagram as Let's Talk Micro, no apostrophe, and I am on Twitter as Let's Talk Micro 1. So please go ahead and follow. I post pictures of organisms, so go ahead and follow. On the last episode, we went over, and when I like to say we, but it's me. I went over tests for gram-negative cocci. I started talking about Moraxella catarrhalis. So when we talk about gram-negative cocci, we typically report it as gram-negative diplococci. So, like I mentioned, it is two cocci together with adjacent sides flattened. They have a kidney or coffee bean shape. I talked about Moraxella catarrhalis, like I said. So, in the textbooks, it is said that Moraxella catarrhalis is part of the respiratory flora. It is primarily seen in otitis media, sinusitis, and pneumonia. And I mentioned that I normally tend to see it in sinus cultures. So the colonies tend to be large and white, and then they slide over the agar when you touch them, which is called the hockey puck. Remember that. And I also mentioned that the ASM, American Society for Microbiology, says that it is classified as a mucosal pathogen. Eventually, it'll be updated on the textbooks. So when I was talking about gram-negative cocci, I went over the catarrhalis disc, right? So what is this test? Well, getting technical, Moraxella catarrhalis, it has an enzyme called butyrate, Butyrate esterase, which releases indoxyl from indoxyl butyrate. This produces an indigo color in the presence of oxygen. Very simple test, very similar to the microdase disc that I talked about for Micrococcus. You place your disc on the slide, and then using a stick, you touch your colonies, and then you put the colonies on the disc, and you observe for a color reaction for 2 minutes. So a positive reaction equals a blue color in 2 minutes. Very simple test. Today, I want to go over tests and biochemicals for other gram-negative cocci. So now that you have the catarrhalis disc down, one of the biochemicals is oxidase. So you remember oxidase, right? If you want a refresher or if you want to learn more, please go ahead and check out episode 10 of this podcast. So Moraxella is oxidase positive, and so is Neisseria gonorrhea and other species of Neisseria. There'll be an episode for Neisseria, so we're just going to briefly touch on it. So let's talk about Neisseria gonorrhea. It is not part of the human flora. It is found during times of infection. It is the leading cause of sexually transmitted diseases, or STDs. Infections may include purulent urethritis, pharyngitis, cervicitis, and conjunctivitis. Then we have Neisseria meningitidis. This one, it colonizes the oropharyngeal and nasopharyngeal mucous membranes. People normally carry the organism without symptoms. And then we have other species of Neisseria that are normal flora of the upper respiratory tract. They can gain access to normally sterile sites, rarely associated with infections. And when these infections do occur, they can include bacteremia, endocarditis, and meningitis. So this group of Neisseria that are normally upper respiratory flora, they are known as NPN. NPN. November, Peter, November. Or non-pathogenic Neisseria. They tend to have a yellow color, and you see them in respiratory samples primarily. I mean, it is upper respiratory flora, So you do see it in respiratory samples primarily. So as we are talking about tests for gram-negative cocci, I'm going to start talking about one that we learned about in school, right? The acid detection test. So getting a little technical and talking about the test, it involves the use of cysteine trypticase agar with 1% of a carbohydrate, of a sugar, glucose, maltose, lactose, and sucrose. And then there's an indicator of fermentation. And can you guess which one? Well, if you're thinking about phenol red, then you're definitely right. So we see our friend phenol red again. So when acid is produced due to fermentation, like we know, the medium, like we have discussed before with the phenol red, is going to change from red to yellow. So different species of Neisseria, they ferment different sugars. For example, Neisseria gonorrhea ferments glucose, Neisseria meningitidis, glucose and maltose, Neisseria lactamica, glucose, lactose, and sucrose. So if you're seeing where I'm going with this, typically when you're a student, the way to remember that was, so you started with Neisseria gonorrhea, and then you had, okay, so you were like Neisseria gonorrhea. So gonorrhea starts with a G, so glucose. It ferments only glucose. And then you move to meningitidis, it starts with an M, so it ferments glucose and maltose. So right, so you start with Neisseria gonorrhea because of the G, only glucose, and then you move on. And then the next species ferments the glucose from the previous species does, plus the one with the letter of the name. So meningitidis, glucose, and maltose. Like I said, Neisseria lactamica, glucose, lactose, and sucrose. So that was the way to remember it. That's the way to remember in school. However, according to the Center for Disease Control and Prevention, which is the CDC, this test is no longer recommended. It is a test for fermentation, so it doesn't work well for oxidative Neisseria species. And then some strains do not grow well. So the bottom line for this is that this test can produce inaccurate results. Some of the species are serious. For example, like Neisseria gonorrhea can have legal implications such as abuse. So we have to make sure that we do produce a good ID. It tends to be an unreliable test, so you want to make sure that you don't identify the wrong species of Neisseria. So what other tests can we use then? Well, here's where molecular plays a big role. We have nucleic acid methods such as the cobas Amplicor and hologic GenProbe Aptima combo. You also have methods like a PCR method such as the GeneXpert for the Cepheid, the CTNG, and you don't have to grow the organisms for these. You collect, if it's typically if it's a male patient, you collect urine. If it's a female patient, you collect urine or a genital swab, and then you place that swab in a special media. I know, like for the Cepheid, it has its own transport media. If it's urine, it has its own urine preservative that it's better to recover an optimal result rather than just testing it directly from the cup, which for the GeneXpert CTNG you can actually test it without adding it to that container. But bottom line is that you don't have to grow the organism. You can just go ahead and test it. And then you typically get results within like 2 hours. You have to keep in mind that depending on the facility that you work on, I haven't had much exposure to the cobas, but I have worked with the GeneXpert with the CTNG test for the GeneXpert. And depending on the facility, you can have the Infinity, which can process about, I think it's like 42 samples. Whereas you can get a regular a regular gene expert, which it can process about 16. Depending on the hospital, sometimes all the modules might not be open, so you can have maybe 4, maybe 8. There's a smaller one that I think can run 4, and the smallest one can run 2. So depending on the facility that you work on, the result might take a little bit longer if you have a smaller instrument, of course. And then there are also cases like, for example, like in this pandemic, where some companies, they started focusing on making tests for COVID, and then the production on other tests was delayed. And then they started being on back order, and that was a big issue with the CT&G for the Cepheid. Like, it used to be in back order because the same type of cartridge that they use, it was the same one that was being used to produce the COVID test. So there were huge delays, and sometimes, large facilities, tests were backed up for days because for this particular platform, it is stable for I think it's 30 to 45 days. So that's a long time where they're getting their supplies. And also another thing to keep in mind is that maybe your hospital is smaller and they have to send their tests to a larger hospital. So transport time, delayed volume, these are all factors. But in optimal conditions, you will get your test results in less than 2 hours. So it's very simple. The patient goes to the hospital with symptoms. A sample is collected. Then it is tested via these methods. So besides, and when I said CT&G, which is for, chlamydia and gonorrhea, Neisseria gonorrhea. So typically they are tested for both. Normally you don't just test for the one. And then you have PCR methods for blood and spinal fluid, where you can check for Neisseria gonorrhea, Neisseria meningitis. So what else do we have besides molecular tests? Well, once the organisms have grown on media, they can be identified using other commercial methods. Of course, such like the Vitek, your MALDI-TOF, Rapid NH, and API NH. There's also an agar that aids in the recovery of Neisseria gonorrhea and meningitis. And these are called MTM and Martin Lewis. So let's start with MTM. What's MTM? Well, MTM is a modified Thayer Martin. It is basically a chocolate agar with some other things added. Well, let's start with why chocolate agar. Well, as you know, chocolate agar, it's an enriched agar, so it helps a lot of organisms to grow better or to grow basically at all, like Haemophilus, with the X and V factors. So not all species of Neisseria, they actually grow on blood agar. The non-pathogenic Neisseria, they do grow on blood agar, and they grow well. Moraxella catarrhalis, as far as gram-negative cocci, grows on blood agar. Meningitis doesn't grow on blood agar, and then Neisseria gonorrhea, it does grow on blood agar, but it grows very weakly. So of course, there's always exceptions, so you can have a strain that might grow a little bit stronger. But typically that's not the case, which makes it challenging, especially when you're on the urine bench, because you don't have chocolate on the urine bench. You have urine cultures is blood and McConkey. So when you have it there and it's growing so weakly, you do your gram stain and you're like, oh, gram-negative diplococci. And you're like, should I call them? Should I not? At that point, just sub it to a chocolate plate and see how it grows. Just to make sure that you have something pure, that you don't have anything mixed, and make sure that it's to see if that's an Neisseria gonorrhea. Also check the patient's history. Make sure that maybe there was a PCR done or for nice gonorrhea and chlamydia. So that will help you support what you're seeing. But bottom line is, it doesn't grow as healthy on blood agar. When you have a source that's genital and you need to rule out Neisseria gonorrhea, you need to have at least a chocolate. And then, depending on the facility, you add an MTM or a Martin Lewis. Maybe you might use both, depending on where you work. So going back to MTM or modified Thayer Martin, so it is an enriched and selective medium for the isolation of Neisseria gonorrhea and Neisseria meningitis. Can you guess what the enrichment portion is? Well, it is the chocolate agar. And then the selective portion is antibiotics. So let's go over them. You have colistin, vancomycin, nystatin, and trimethoprim. So maybe you students out there, can you guess? Microbiologists, do you know? Well, colistin, it is used to inhibit gram-negative bacteria. Vancomycin, of course, gram-positive bacteria. That's what you use to treat like MRSA or methicillin-resistant Staphylococcus aureus. Nystatin is to inhibit yeast, and trimethoprim is to inhibit the swarming of Proteus. Proteus is not going to swarm, but however, technically it can grow on MTM because Proteus and its family, like Morganella, Providencia, they are intrinsically resistant to colistin. And what's an intrinsic resistance? It's a natural resistance that they have. So you cannot treat the organism with that antibiotic because it's naturally resistant to it. Like, let's say, like Klebsiella being resistant to ampicillin. So that's what you have for modified Thayer Martin. So keep in mind that, like I like to say, these organisms, they do not go to school. So you can have some growth. So they're selective for Neisseria gonorrhea and meningitis, but you can have other organisms maybe grow to a lesser extent, of course, but they can. And one thing, as I was teaching and I saw that Moraxella catarrhalis actually grows well. And then we have also Martin Lewis. So Martin Lewis is a modification of Thayer Martin. So what's in here? What's the modification? Well, instead of nystatin, there is ansamycin. And also there is a higher concentration of vancomycin. So this is used for the selection and isolation of Neisseria gonorrhea. So you're on the processing area. You receive a sample of genital source. Typically, of course, already from the setup, if you have like a gonorrhea culture, you can have a chocolate and a Martin Lewis, or a chocolate and an MTM, depending on the facility. But let's say if they order a wound culture, but the source is genital, then you will go ahead and add a Martin Lewis or an MTM, because these things are not set in stone. So you have your provider's orders a gonorrhea culture from any source, and then of course you set up your chocolate, you set up your Martin Lewis or your MTM. But sometimes they order a regular wound culture, which the standard setup is blood chocolate McConkey, maybe BEA, anaerobes if the source indicates it. But then they put let's say the wound culture and then source is vaginal or source is genital. So in that case, you will do your standard setup, blood chocolate McConkey, and then you will add a Martin Lewis, just to make sure that you rule out Neisseria, especially considering the source. If you have a vaginal source, normally you have Enterobacterialis as part of the flora. So maybe that McConkey, that McConkey can get pretty full sometimes. And then you have your chocolate plate. There's a lot of genital flora, so it can grow, a lot of cocci. By using this plate, you're giving that Neisseria gonorrhea, if it's there, a better chance of recovery. So that's what you will use it. So based on the source. So this is one of those things that you always have to use your judgment and your knowledge to make decisions like this. You're like, okay, it's not a gonorrhea culture. This typical setup doesn't have this particular plate, but you do add it. And that's something that's very normal in the microbiology field. But I know like sometimes that might differ from other areas of the lab. Basically, other areas like in the core lab, you have your SOPs, you follow that procedure. And then with micro, we have our procedures for different cultures. But then if a source, it's something where that there's this pathogen, and that typical culture setup doesn't have that agar, then you go ahead and add it just to make sure that you give that organism an optimal chance to grow. Like I said, a genital source, many bacteria there. So by throwing in that Martin Lewis or that MTM, you're giving that organism not only a chance, but then you are also successfully ruling it out. And that way you don't miss it. And of course, this is always about the patient. So we make sure that we send out there, the best information. So now that we have learned about 2 more agars that are selective. So in previous episodes, we talk about McConkey, selective for gram-negative rods, differential for lactose fermentation. We talked about hectone, XLD, selective for gram-negative rods, differential for lactose fermentation and H2S production for hectone and XLD. And then for this one, it's selective for Neisseria gonorrhea on Martin Lewis, and then modified Thayer Martin for Neisseria gonorrhea and Neisseria meningitis. So there you have 2 more sets of agar that you learn. So now, knowing that, you microbiologists, I tell you this. You have a McConkey, you have a blood, you have a chocolate, you have a Martin Lewis, and you have a PEA. Does it matter the order that you streak them? Well, we have learned that it does, right? The order of streaking is always from least selective to most selective, because you want to make sure that you don't transfer any of that selective material to the next plate, potentially inhibiting the growth of the organisms. So given those plates, what's the order of streaking? Remember, always from least selective to most selective. So of course you will have blood, chocolate, PEA, McConkey, and then Martin Lewis, an MTM, or either or. But blood, chocolate, PEA, McConkey, and Martin Lewis or MTM. So just to recap, in addition to the Cataralis disc, which is helpful for Moraxella cataralis, we use oxidase for gram-negative cocci or diplococci. And then here, molecular tests, they play a big role. Like typically for gonorrhea, you do a PCR. And then also mostly for genital sources. And then for other sources, providers like to order a culture. So in gonorrhea, we can, in the bacteriology side, we can go ahead and process it. Like I said, a chocolate, a Martin Lewis, and basically any source we can perform that culture on. So molecular cultures, and of course, you have these 2 plates that will help in the isolation of Neisseria gonorrhea and Neisseria meningitis. Because the other Neisserias, the NPN, they're normally normal respiratory flora, but at the same time, they tend to grow in regular blood and chocolate agar, and they grow well. So there you have it. Some information on gram-negative cocci. And that, my dear audience, it's the end of this episode. I hope you enjoyed learning about tests and biochemicals for gram-negative diplococci. I sure enjoyed talking about them. Like always, please continue staying safe, continue staying motivated, bring that passion into work. Just be passionate, produce the best results you can. That way you have the best outcome for your patients. But such an important work, it's best to do it motivated. That way you can get through the piles of plates, and like I said, produce the best results. So please stay safe, stay motivated, and of course, continue talking micro. Until the next time, bye.